So I see 3:30 on my computer. So welcome back everybody to the Evolutionary Ecology session. I think you've probably seen this enough times, but the way that I'll do things is when there's about 3 minutes left, I will bring up that green check. And when there's about 1 minute left, I'll bring up the red X. And at that part where there's about 1 minute left, if there's some Q&A, if the current speaker takes down their slide, stop sharing so that the next one can get them up and running, we'll be able to keep this moving. So I'm not going to waste any time now. Our first speaker in this section is Vandana Gupta. Vandana.
Hello. Okay, I'll start sharing my screen. Cool. So you can all see my screen, I hope. Okay, so hello everyone. I'm Vandana Gupta. Am I clearly audible to everyone? You can understand everything? Hello?
Yes, everything works.
Yeah, okay, cool. So I'm just presenting my PhD topic here. I'm a third year PhD and I'll be presenting different things we have been working on and looking at. Okay. So starting out with parasites. So parasites is not a new term which I need to introduce here. Parasites range from bacteria, fungi, different kind of are invertebrates which parasitize a lot of hosts. But the thing about parasites is they never are parasitizing a host alone. So whenever you see a host, there are a lot of parasites and co-infections, be it humans, like we don't even know like how many strains and different kind of parasites are present on our body right now. So all kind of hosts are parasitoids, and there are different factors which affect these co-infections, like our environment, primarily what kind of environment we are in, our genetic structure, nutrition, vectors. So all these things affect co-infections, and co-infections in turn, they affect all the host parasite interactions and the evolution of virulence. For me, when I'm studying co-infections, what I'm focusing on is how parasites are interacting with each other, how parasites are interacting with other parasites when they are in co-infection in a host. So during co-infections, parasites can have positive, negative, or no interactions among them. So in this diagram, you'll see the three interactions here where at least one of the parasite is getting benefited from the association. They come under an umbrella term, facilitation. So basically positive interactions between two parasites, which I'm interested in. So in my case, when I'm studying co-infections, my host is a tomato plant, Solanum lycopersicum. We have two parasites in the lab we are studying. My first parasite is a spider mite. Spider mites are called spider mites because they make heavy webbing onto the plants they are eating from. So that's why they're called spider mites. And the second one is tomato spotted wilt virus, which is a plant virus. And it's in nature, tomato spotted wilt virus, it is exclusively transmitted by thrips. But one thing to notice here is this virus can be easily transmitted mechanically in the lab, which we are doing. So in the lab, I'm not employing any thrips. We may inoculate our plants mechanically ourselves without any thrips. So for now, my PhD thesis, the question was, so how does a plant virus and another parasite, which in my case is spider mites, how are they interacting with each other when they are infecting the same tomato host? And so just to mention here also that I told you that this virus is transmitted exclusively by thrips. So when I'm studying tomato spotted wilt virus and spider mites, I'm studying virus and a non-vector herbivore fitness. So my question is, does virus infection always change non-vector herbivore fitness? Because for virus and vector interaction, there is a whole lot of literature which studies how the transmission and everything is changed. But the literature about virus and non-vector is not so blurred because there are different directions in this. So, but anyhow, there are some studies which have already tried to get into this. So one of these I'm showing here is the study on the same system what I'm working on is tomato spotted wilt virus and Tetranicus urticae. So in this study, they showed that the plants in black, which are infected with viruses, on such plants, spider mites have more number of juveniles produced than the mock plants. Now, another study also with the tomato spotted wilt virus and the white fly, this showed that when the plants were infected with thrips, which transmit the virus, on such plants, there were more number of eggs produced than the mock plants. So there have been studies which showed that the virus do in fact actually increase the fitness of their known vectors. But there are equal number of studies which showed negative or no effect also. So we don't really have a definite direction in these kind of interactions. To tell you a bit more about the system I'm working on, so my first spider mite is Tetranicus urticae, and it is a journalist herbivore. So this herbivore is infecting over thousands of species of agricultural importance. I just have one question. Can you see my full screen? Because for me, I have this.
I think we can see your full screen.
Okay, perfect. So Tetranicus urtica is a journalist herbivore and is infecting over thousands of species of agricultural importance. It's a haplodiploid, so the females are produced when the egg is fertilized by a sperm. And in this figure, to tell you, the bigger one is the female spider mite. So it's very visibly different from the male one. It's bigger and the abdomen has a different shape than the male. My second spider mite is Tetranicus evansi. The difference is this one, Tetranicus evansi, it's a specialist herbivore on Solanaceae family and also bean. This spreads very fast compared to T. urticae. It's red in color, so it's visibly distinguishable. And but the life cycle is same. They both have around 14-day life cycle in the lab, starting with egg, larva, 2 nymph stages, and the adult. Now talking about my virus, it's tomato spotted wilt virus. It belongs to a genus Tospovirus of viruses. It is transmitted exclusively by a few species of thrips in nature. And like the spider mite, it leads to huge economic losses all over the world, throughout the globe, and also especially in the Mediterranean region. The crops are, it leads to like billions of loss. And the visible symptoms are, as you see in this figure, it leads to bronzing of leaves. So the plants are very stunted. There are necrotic lesions onto the plant, and the leaves eventually becomes brown and bronzed. Okay. So in my first year, we started with the experiments and question, does the virus, tomato spotted wilt virus, will have a positive effect on the life history trait of spider mites? And also to tell you about, like I told you, we do mechanical inoculation of the plant. So how does that work? So it's very simple. So for this, we take some leaves which are already been infected with viruses. So it could be frozen leaves. It works like you can freeze it in -80 degrees, or it can be a live plant. So we take some leaves, some buffered charcoal, we make a paste out of it. And in this paste, we add carborundum, which is a surface abrasive, so that the plant surface can soak the solution well. So using this surface abrasive, we then rub the solution onto the leaf of the plant. So you have to like manually put the solution onto the plant, and the plant absorb the viruses. Now talking about the experiments, how do we do our experiments? So we start with mechanically inoculating the plant with virus using the solution. As I told, we rub the solution onto the first leaf of the plant, infect it. Those plants are left like this for 14 days. After 14 days, we check if the plant we put the infection on, is the plant infected or not. And when the plant is infected, there are two ways we can do our infections. So for a lot of my experiments in first year, I did experiments on cut leaves, which were unsuccessful. I'm not showing any results here, but it's one of the way people do that. And the second way is we do our experiments on the whole plants. So we take the whole plants which has been infected, we transfer the spider mites onto the plant, and we isolate the spider mites onto the leaves using a leaf cages we made with the muslin bags. This is how the setup looks in both conditions, with the cut leaves and the whole plants. And also, so for when we transfer the mites on day 14th, they lay eggs, which we can count. And on day 28, spider mites will complete one life cycle. So after one generation, we can count how many mites have been produced on our control plants and the virus infected plants to see the effect of virus onto their reproductive rates. So this is the result from my first experiment, which we did on whole plants, and it was the first experiment which was successful in our case, because on cut leaves, the experiments just didn't work. So on the x-axis, you will see its spider mite species. It's the are two spider mites, Tetranicus evansi and Tetranicus urticae. And we are counting the total number of adults in both the spider mites. On the top, I have Microtome and Moneymaker. These are two different varieties of tomato. These I have used because we wanted to see which variety we wanted to continue on for the experiment. So we just employed two different varieties of tomato. So the gray bars here are the mock plants, and the black one here are the virus infected plants. Yeah, I should have mentioned. In this experiment we had very interesting results because we see facilitation in both the varieties of plants in both the spider mite species. You see, for both the Tetranicus evansi and Tetranicus urticae, we saw that there were more number of adults produced in the plants which were infected with viruses in all of the cases. So we saw facilitation on both the mites. And the positive effect was seen as early as 4 days after introducing the mites. So these results I'm showing you are for total number of adults which are produced after 14 days. But in a separate pilot experiment, I also counted the number of eggs on another experiments. And I also saw that in virus infected plants, there are more number of eggs produced onto the plants than the mock plants. Now, coming on to the effect of spider mites on the tomato spotted wilt virus. So in the first part of this, I studied the effect of virus onto the spider mites, but we have to see if the spider mites are also affecting the virus in some way. So these experiments we did last year. So in these experiments, we employed Q-ELISA to measure the amount of virus titer in the plants. So in this plant, we had 4 different time points, day 6, 13, 20, 27 after the virus infections. And as I told you, we put spider mites on the day 14th. So I have 4 time points for the plants with viruses, and I have 2 time points with co-infections. So we wanted to compare the co-infection treatments and just virus treatment to see if there is an effect of spider mites onto the viruses. So these are the results of that experiment. So on the left, it's number of adults per centimeter. So because in this experiment, the viruses, they have a huge impact onto the host height. So to take into account that, it's number of adults per centimeter height of the plant. On the x-axis, there's plant treatment, France 81, Live 1137 mock. So these two are just the 2 strains of the same virus. So the same tomato spotted wilt virus, both of them, they are 2 different strains of the same virus. So in this experiment, I saw that in the first strain, France 81 strain, there was facilitation. There were much more number of adults than in the mock treatment, but not in this second strain. So I saw facilitation only with France 81. Now, when you look at the graph on right, its log relative concentration is the virus titer of the plants, virus titer in the plants, and days after infection. So the dotted lines are the plants which are without mites, so only single virus treatments. So they have 7, 14, 21, 28. I have 4 time points with only viruses, and I have 2 time points with co-infections, which are these solid lines. The red one is almost mixed with the dotted one. So the solid lines are the co-infection treatments on day 21 and 28. So interestingly, in this experiment, I saw that the France 81, the red line, the first strain, consistently from day 7 to day 28, there was more amount of virus, like there was more virus titer in this strain compared to the another strain, and that corresponds to the facilitation we see on the left. So the more the amount of facilitation, more number of adults produced onto the plant, it's actually corresponded with the amount of virus that plant is having. But the main question was, will I see any effect in the dotted lines and the solid lines, which I didn't see. So on day 21 and 28, we didn't see any difference between the presence of the plants where there was co-infection or just the dotted line virus infection. So in this experiment, I didn't see any effect of co-infections onto the virus fitness. So there was no effect on the Tetranicus urticae on virus load. There was indeed a positive effect of viruses on Tetranicus, and host phenotypes differ as per the treatments, but irrespective of co-infections. So there was a huge difference across different host phenotypes when I measured because of different viruses, but it had nothing to do with co-infection. So spider mite did not affect any aspect of the virus fitness and also on the host. Now I'll go firstly a bit in the immune system. So when I'm talking about the spider mites and virus, how they are impacting each other. So they are interacting with each other. The viruses and spider mites are interacting with each other definitely, and it could either be via host immune responses indirectly, or it could be a resource-based interaction like free amino acids. So a lot of studies have confirmed now that the presence of plant viruses into the plant, it leads to more number of free amino acids released in the plant, which other organisms like herbivores can consume and use. So talking about plant defenses. So when spider mites
Sorry,
infect,
Donna, I
Oui.
have to interrupt you. We're at the 15-minute mark.
okay.
It happens. If you want to, do you have a final acknowledgements?
Yeah,
like
I'll
that?
try to say.
And then if there are questions, then Donna, folks can put them into the chat.
Yeah, I'll just try to just wrap it up in 2 minutes. So what I'm basically saying is when both of the parasites are attacking, they are attacking different chains of plant defenses. They are opposite chains. So I was going to present another experiment where I altered different timings of spider mites, but
We have to move on. I'm sorry.
yeah. No, it's fine. Okay. No, yeah. So eventually I didn't see any effect of spider mites onto the virus till now, but a positive effect of virus onto the mites. I could, yeah, conclude that. And yeah.
Right, thank you. Alexi, can we get your slides loaded?
Okay, so I will stop sharing. Think.
All right, and I will start sharing.
Thanks.
And hopefully you can see everything.
Good.
Okay. Hi, everyone. My name is Alexi. I'm a PhD student at Old Dominion University, and I'm really glad to present this small model I wrote to illustrate the concept of trait-based environmental filtering. And my dissertation is mostly about avian communities and how they change along an urban gradient. But hopefully by the end of this presentation, I can convince you that this model can be applied to different communities and different gradients. And it all starts with the simple observations that biological communities generally differ along the gradient. And one of the possible reasons for that is the environmental filtering. So in the system I work in, it has been observed that urban species usually have specific sets of traits, and probably it's because this urban environment filters the species from the regional pool based on some traits that they might have. And therefore, the local community that we can see is a product of several processes. First, of the dispersal from the regional species pool into the habitat. And then, maybe the habitat can be unfavorable for the species. If it is favorable, then still there might be some organisms already present in the system, and therefore there can be some competition or some other inter or intraspecific interactions. And all of these ideas led me to develop this agent-based model. So it has two main parts. First, there is some meta community or regional pool. Just for simplicity, I will use them interchangeably, and the local habitat. And because it's agent-based model, it operates at the level of individuals, and it all starts from the agents dispersing from the meta community into some local habitat. The meta community itself, just to make everything more or less understandable, is portrayed as a data frame with species, their abundances, and trait values. And the local habitat itself is composed of a set of many cells, just to allow for habitat heterogeneity. And then all these agents can disperse from the meta community into cells and then between cells. And inside of the cells, something happens that we go through later. To start with the meta community, again, this is some data frame with abundances and traits, but what is the relationship between traits and abundances of species? And as an inspiration, I use this work of Hoffel and colleagues that suggests that the distribution of traits across species and communities kind of resembles the normal distribution. And the distribution of traits across individuals, irrespective of species, is more similar to Cauchy distribution, which also looks like a bell curve, but a bit more narrow. And then what is happening inside of the local environment itself? Again, I took this paper of Muhammad et al. As an inspiration that suggested that local environment can be portrayed as a unidimensional row of cells. And in my case, the question was, okay, we have many, many cells, and there is some environmental factor at each cell. How does it depend on the cell location? So it can be random, can be clustered based on the position of the cell, can be correlated with the position of the cell, or can create some very nice linear gradient. And then again, there is the recruitment of agents from the meta community into the local cells, but also I would allow the agents to travel from cell to cell. And both of these processes were modeled as a Poisson process. And the main part is whatever happens inside of each cell. So once we have a cell with some environmental factor level, with some resource, and some agents ending up in these cells, they need to consume resources. And the amount of resources consumed is pretty much how the environmental filtering is portrayed in this model. There are some calculations, but to make everything simpler, there is a term for environmental filtering or clustering of those traits, so that there is some environmental factor level inside of a cell, and it has some traits. And just to make everything simple, those traits and environmental filter have the same units, same scale. So the local cell will preferably filter for the individuals that have a trait value close to the environmental level in the cell. And that would result in some niche clustering. But we know from theoretical ecology that species with, or individuals with very same trait values cannot coexist because there will be some competition. And therefore, there's also a term for the competition or niche dispersion. And another thing is, even if the individual ends up in a cell with particularly favorable conditions, if there is no resources, there is nothing to eat. So this competition term will take a form of either 1 or 0 based on the resource availability and whether there is a high competition with other individuals that are already present in the cell. And then why to consume the resource? Well, because we need to reproduce based on this resource. So both all individuals will have some maximum lifespan upon reaching which they will be just eliminated from the population. And each individuals will be assigned some body mass for reproduction, after reaching which they will just split into 2 individuals with body mass adding up to the parent body mass and the trait value derived from the parent plus some random error. And both of these values were drawn for all individuals, irrespective of species, so it's more or less neutral process. Maximum lifespan was modeled as an exponential distribution, and then body mass at reproduction was a logit-like function. So just to overview whatever happens in one time step, first, all the agents are recruited from the meta community into the local cells. All of them are agent by one time step. Then mortality happens, reproduction happens, agents travel to neighboring cells with this cell-to-cell dispersion, and then we go through this complicated process of the resource consumption. But before we can initiate the model, we need to create the meta community somehow, and we need more or less realistic relationship between trait values and species abundances. So to do that, I just modeled some space of these 2 parameters of Cauchy shape and a normal distribution shape, and try to pick such a combination of these parameters that would generate a species abundance curve the most similar to whatever we observe in reality. And just an example of reality, I took the estimates of bird species in southeastern coastal plain in the US. And I ended up with some combination of values that was the best among all these combinations, with pretty normal shape of the Cauchy function and very wide normal distribution of trait values across species. And I just plugged it in and let the model run. It was running for 500 steps with different combinations of this special configuration of environmental levels, with different combinations of cell-to-cell dispersal and recruitment from the meta community. And the result was pretty much what we would expect from theoretical ecology. So this trait structure of local community is some kind of product of whatever trait distribution is in the meta community and what is the environment filters for. And the balance between these two processes is governed by the recruitment rate from the meta community into the local environment. So if the recruitment rate is high, then the trait distribution of the local community will mostly resemble the meta community itself. But then if there is limited recruitment, then the trait distribution of the local community will be more or less independent. When we look at the species composition and its dynamics over time, it's pretty much the same picture. So when there is a low recruitment rate, then there is some core of species that could establish local viable populations, and it just goes on. And there are a little bit of transient species, but they don't survive well. But when we increase the recruitment rate, then there are more and more transient species, and they come and go. But at any point in time, we will see very high species richness. So that's what we see with observed taxonomic diversity in the outputs of the model. Whenever there is a high recruitment rate from the meta community, because of all the transient species, the observed diversity is quite high. And the same pattern will be seen in different measures of taxonomic diversity, irrespectively of the spatial configuration of environmental levels across cells. But then dispersal from cell to cell doesn't affect this, not that much. So you would say, well, yes, this model pretty much shows whatever we already know from theoretical ecology. And yes, that's true. But why I developed it? Because it could be modified to address different questions. So for example, as it is, the reproduction is modeled as pretty much a sexual process. What if we modified it to allow for some sexual process, maybe for some allele effects and things like that? We can modify the hereditary mechanisms in the model as well. And both of these things could help us to address different eco-evolutionary dynamics in communities. But for particular interest for me, $1 million question in functional ecology is what traits to use when we look at environmental filtering, because there are so many of them. You can describe any organism in an infinite number of traits. And once you do that, you have to deal with this multidimensionality in the data, which can be very tricky and very hard. So what if we could use some method to figure out what traits of organisms are the most influential for environmental filtering? And in my model, we have a trait that we are 100% sure is responsible for the environmental filtering. So it could be used for testing potential methods of figuring out the most relevant traits. And maybe people in the audience could think of some other ways that kind of model can be used. And this, I would like to thank all the colleagues that helped me to make this model more deliverable. And I would love to take any questions, either in chat or you can just email me if you want. And thanks.
All right, thanks, Alexei. We actually do have a minute or so for questions if anybody has one. Looking, anything in the chat? If you have a question, please, if it comes to you, post it in the chat. And to be there for Alexi. Thanks.
Thanks.
Okay. Rebecca, looks like you got your slides loaded. So if you're ready to start, go ahead.
Sorry, I was trapped in this situation where am I still sharing my screen? No?
No, you lost it. It was
Okay.
up.
Yeah, I was trapped in a situation where I couldn't unmute while my screen was sharing because I couldn't get access. Okay, we're going back in. Sorry about that. Okie dokie. All right, well, thank you for coming today. And if you're watching this recording later, thank you also, you still count. I'm going to blast right in because I know my talk is going to cover all kinds of different things, but I'm excited to tell you about some of this research I've been doing primarily on this species that's here in the background. So I like to start with a picture like this, a little photo collage, because as evolutionary biologists, a lot of us are trying to understand diversity and the wide range of...
Sorry, it seems like your slides might not be sharing correctly. I don't know if other people are seeing this too.
Yeah, I'm also seeing it. It was there and then it disappeared.
Yeah, it was kind of like flashing for some reason.
Yeah.
Wow, we're doing great. Is it working now?
It keeps flashing between like the presentation view and
Let
showing.
me try this. I'm going to start sharing one more time without an extra monitor.
I think that'll help probably.
Yeah, sorry about that. Can we just do this? Does that work?
It looks good.
Okay, I'm gonna save time and just say carotenoid pigmentation in birds. That's what I study. So most of the red, orange, and yellow colors in birds are formed from carotenoid pigments. And I'm not the first one to have explored these. So I'd like to generalize perhaps overgeneralize a very large literature spanning decades to say that one reason these colorful traits in birds are intriguing is that increased expression of carotenoid-based coloration tends to be linked to measures of improved quality. So things like fewer parasites and improved immune response and so on. And more colorful males are often preferred by females and vice versa. So carotenoid-based colors in birds have become a somewhat textbook example of a condition-dependent signal of quality, where they appear to often communicate information about a prospective mate's ability to be an excellent mate and father to offspring. But the natural next question here is, why should ornamental coloration have anything to do with these other aspects of quality? Why should something as arbitrary to us as the deposition of pigment in a feather have anything to do with, say, the ability to perform an immune response? So this has been the focus of my research, and to begin to understand this, we have to take a look at what it takes to become a colorful bird in the first place. So carotenoid pigments, one of the reasons that they're interesting is that unlike other pigments that birds will use to color their integument, they cannot be synthesized entirely from scratch in the bird's body. So birds can only acquire carotenoids from their diet. Now, fortunately, plants and insects tend to be quite rich in carotenoids. So this house finch will eat some seeds, obtain this pool of carotenoid resources that he can absorb into his gut and circulate around, put into different tissues, ultimately potentially deposit some of that into his ornamental trait, in this case, feather coloration, or retain them in the body. Some carotenoids, for example, are precursors to vitamin A, so that's an alternative use. So we've been looking at these sorts of pathways and trying to understand this link between coloration and quality. And a step that recently we've become more interested in is one that essentially every red carotenoid colored bird must go through, and many yellow birds as well, where almost all of the carotenoid pigments that are available in plant and insect matter that these birds are eating are yellow, and they must be metabolically converted into red forms before being deposited in the ornament in these species. So this is interesting because it gives us some new hypotheses for why carotenoid-based coloration may be condition dependent. Like maybe some higher quality birds are redder because they're better at performing this conversion. And that's actually sort of, well, my colleague a few years ago performed this meta-analysis to kind of look at this relationship between carotenoid metabolism and condition dependence of coloration. And he found that essentially birds that were using metabolized carotenoid pigments in their feathers versus birds that used just dietary pigments in their feathers communicated more information with their color. There was a stronger link to quality in birds using metabolized carotenoids. So this is interesting to explore further, but it's been difficult because until quite recently, this actual metabolic conversion step was basically a black box. But fortunately, within the past 10 years, we've had these enormous genetic breakthroughs that have allowed us to finally isolate the enzymes that appear capable of converting dietary yellow to ornamental red pigments. These two enzymes that we know today are CYP2J19 and BDH1L or BDH1-like. So here's the full names in the corner if you're interested. And they both work together in a stepwise process to produce these metabolized red ornamental carotenoids from dietary yellow precursors. And just as a quick note, either enzyme on its own will actually act on these dietary yellow carotenoids, but it will just produce a different yellow carotenoid. So both are required together to produce these red pigments. So if we come back here, this is pretty exciting because perhaps we can finally open that box and we know some enzymes that might be able to do this yellow to red conversion. And we could potentially leverage this system to better understand how yellow to red carotenoid metabolism may help us better understand why red coloration could link to these aspects of quality. So this leads me to my study. I was interested in exploring these questions further, and the system in which I chose to do this was the house finch. Now, they've been quite well studied within the field of carotenoid coloration for many of the same reasons that I think they're a great model to test these questions of conversion. House finches are unusual among really any of the bird species worldwide for the most part, in that they show a very striking amount of variation in hue. So individuals within the same population or even the same individuals across different seasons of molt can range from dull yellow, like on the left here, to brilliant red. And studies have shown that these dull yellow birds are not performing that carotenoid metabolism step, and they're only depositing dietary carotenoids in their feathers. Whereas the brilliant red birds are producing tons of metabolized red carotenoids, specifically one called 3-hydroxyechinonome, and that's where they get their coloration. So if I'm looking at really the effects of carotenoid metabolism on relationships between color and quality, this seems like a great system to do it. But to start out, I need to see, are house finches using this CYP2J19 BDH1-like system that's been found in other taxa of birds to produce these red carotenoids with which they color their feathers? And I've labeled this prediction 1 because there are, of course, downstream predictions. I want to ultimately link these to questions of relationships between ornamental coloration and quality. So, for example, my second prediction is that the functionality of that 2-enzyme system might be sensitive to subcellular conditions, which in turn would help create this link. But that's really a whole can of worms that I'm not going to open today. So we'll start with focusing on prediction 1. And this has 2 major components. First, I want to see, do house finches express these 2 genes in one or more of the tissues actually related to ornamentation? And second, can this 2-enzyme system produce the pigment that the house finches are actually using? Can they make 3-hydroxyechinonome? So to test this first prediction, I wanted to see, do house finches express these 2 genes at all? So to do that, we needed some birds. This is a picture of Jeff Hill in Alabama capturing some house finches. So we got samples of the retina, follicles, and livers of a bunch of molting male house finches from Alabama. The retina is kind of a control tissue because we know that most birds, well, the birds that we've studied to date, use these 2 genes to produce red carotenoid pigments that are involved in their vision. And then the liver and the growing feather follicles are the 2 main tissues that have been proposed to be the main sites of carotenoid metabolism. So we extracted RNA and looked at gene expression across these tissues. So here's a way we can visualize patterns of gene expression, where I'm showing you right now, just basically we're zoomed in on the region of the house finch genome that contains the gene CYP2J19. We're looking at retinal data. So each of these rows is a sample from a different individual. And then each of these gray boxes near the top is an exon, or really a protein coding region of the CYP2J19 gene. So when you're looking at this, basically, if you see more color, that means more expression. So we do see CYP2D19 expressed in the retina, where it likely plays a role in making pigments for vision, but we don't see it in the livers or follicles of male house finches. This is in contrast to BDH1-like, which is expressed both in the retina and in the follicle. But we know that BDH1-like alone cannot produce these red pigments that house finches are using. Instead, they modify yellow pigments. They actually make canary xanthophylls for most precursors. So we actually don't find support for our first sort of sub-prediction here. House finches do not appear to express both genes in the tissues actually related to ornamentation. But I still think it's worth exploring prediction 1B because it's also possible. I only looked at 2 potentially ornamental related tissues here. Maybe there's some other tissue or location that I'm missing. Maybe something happened with the RNA sequencing. Who knows, right? So let's just see, can CYP2D19 and BDH1-like together produce the main pigment that house finches deposit in their feathers, 3-hydroxyechinanone? So to answer this question, we relied on cell culture in our enzymatic activity assays. So for this, we cloned the 2 genes from house finch cDNA, and we basically insert those genes into a plasmid that contains a promoter and then a fluorescent protein that we can use to verify that it's being expressed. We transfect these genes into our stable cell lines that we keep in the lab, and then we can feed these cells that are expressing these genes, one or both of them, different yellow carotenoid substrates. And then after some hours, we can collect the cells, extract carotenoids, and see what they were able to make. So essentially, this allows us to create a complete map of the BDH1L and CYP2D19 system, what it can produce from these 4 main dietary carotenoids that birds are accessing from their diet. And here's that map. We found that this 2-enzyme system that previously had largely been tested in chicken genes, it can produce a bunch of different modified yellow carotenoids like those used in American goldfinches, and a bunch of different modified red carotenoids like those used in northern cardinals. But importantly, we never detected the main house finch pigment, 3-hydroxyechinanone, or any pigments with similar structures to 3-hydroxyechinanone in any of our assays with any of our precursors of gene combinations. So we also don't find support for the second half of this prediction. So house finches don't appear to use CYP2D19 and BDH1-like to produce the red carotenoid pigmentation, which is surprising because evidence so far has been quite consistent with birds using this 2-gene system. So very briefly, let's put this in a broader evolutionary context. So here I've put up some somewhat terrifying, a series of carotenoid structures. That's if anyone out there is a carotenoid nerd. But essentially what we did is we surveyed the literature and we recorded whenever a carotenoid of a particular structural class basically was reported to be present in a particular bird's feathers. So here we can focus in just on the red colored boxes here essentially, because those are carotenoids that are like the ones that are present in house finches. We map this on a phylogeny of bird taxa, and we can see the house finches here. Well, I'm showing you that the house finch is here, but what's interesting is we not only see these red boxes signifying 3-hydroxyechinanone and similar carotenoids in the house finch and its closest finch relatives, like here's the red crossbill, but also in some quite disparate species like Theropidae tanagers, and my computer is opening OneDrive. There we go. Also some much more unrelated species like golden crowned kinglets. So this is really interesting because we've just shown that CYP2D19 and BDH1-like cannot produce the types of carotenoids that birds like house finches and these other birds are using. So there must be at least one alternative pathway that birds use besides the one that we've discovered to produce these pigments. I think this is especially interesting because if we're trying to draw conclusions by looking at this diversity of birds in the wild, it's important for us to know how they're actually making those colors, because I think that could have quite strong effects on their condition dependence and the sort of information they're communicating. So with that, I'm happy to take questions in this platform or over email. And I'm approaching manuscript submission somewhat asymptotically, but look forward to seeing it soon if you've got any more questions. Thank you.
All right, thanks, Rebecca. Yeah, we have a minute or so for questions. Is that Hagar? Do you have your hand up? Oh, that's clapping. Sorry.
Thank you.
I'm going to ask a quick one, and I don't know if you can say it here, but the system that you're looking at, do you think that it lends itself potentially to sort of dishonest signaling?
I don't think so in the house finches in particular. This is getting into a bit of that speculation, but I think that the enzymatic system that they're using is probably one reason that they vary so much more in hue than other birds. I think that their system is especially sensitive to whatever kind of variation. It's basically sensitive and doesn't work if something's wrong. So I think that house finches might have this very kind of quick and dirty way of making red, and a lot of them it's not working out, and that's why you see so many yellow birds. So I don't expect to see it here, but it'll be interesting to see if there's like different costs or benefits to using one pathway or another pathway. Maybe some birds can use either, some birds use both. House finches seem to use kind of just the one. So that's something I would love to learn more about.
Okay, thanks.
Yeah, thank you.
And I think it's at this point we have a little bit of a short session today. So Pauline, are you here? Are you able to get your slides loaded?